Biomics. In this today's video, we are going to discuss about the amino acid tRNA synthetase. So this tRNA and amino acid, they are working in the formation of protein. So before that, this amino acid and tRNA needs to be activated, and it is being activated by an enzyme which is known as aminoacyl tRNA synthetase. So what happens when there is a amino acid? This amino acid is having a amino group, R group and a carboxylic group. Near the carboxylic group, this oxygen is having a negative charge due to lone pair of electrons. For its activation or aminoacylation of amino acid, it requires a ATP, and this oxygen which is having a negative charge, it attacks with the alpha phosphate of ATP, and during this time there is release of pyrophosphate. That means 2 phosphate are released and it forms the adenylated amino acid. Okay, so here we can see that in this diagram, this enzyme aminoacid tRNA synthetase, it is having 2 domains. In one domain comes and bind the amino acid, and in the other domain comes and bind the ATP. And here this reaction takes place. This amino acid is having a reaction with this alpha phosphate of ATP, and that is release of pyrophosphate. Here we can see. In this way, adenylated amino acid is formed. What next? After the formation of this adenylated amino acid, it binds with the tRNA. This adenylated amino acid now comes and binds with this tRNA. In case of tRNA, in the 3' end of the tRNA, it is having a CCA sequence. And A means adenine. In this adenine is having a 3' OH group as well as 2' OH group is present in it. This both have the ability to make a ester bond. Okay, when this 3' OH group is present, that means the class 2 tRNA synthetase is working here. If 2' OH group is present, that means class 1 tRNA synthetase is working. If it is 2' OH group, it attacks with this carboxylic part of this adenylated amino acid, and it creates a bond. This bond is known as ester bond. Okay, after this reaction, there is release of AMP. AMP is released, and now this adenylated amino acid which was activated, and now it is transferred into the tRNA. Here we can see that here there is activation of this adenylated amino acid, and now it is being transferred into this tRNA, which is known as aminoacyl tRNA. In this way, the tRNA synthetase works. Here we can see that one amino acid is present. There are 20 different type of amino acid, and for each amino acid there are 20 tRNAs present. This amino acid and tRNA are specific for each other. This first amino acid, it works with ATP, and in presence of aminoacid tRNA synthetase, it releases pyrophosphate. So here it forms the acylated amino acid. Then it binds the specific tRNA by releasing AMP. This AMP is released. Now it binds in the CCA region of the 3' of the tRNA. In the 3' region of tRNA, CCA sequence is present, and near the A, this amino acid is coming and binding, which is known as charged tRNA, and the bond which is formed here is known as ester bond. In this way, the work of aminoacylation of tRNA is taking place. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.